Hello everyone, welcome to Infinity Learn by Sri Chaitanya. This is Dr. Priyanka and today we are going to discuss a very important topic of the chapter biomolecules. जैसे हमें पता है biomolecules एक बहुत ही ज़्यादा important chapter है grade 11th zoology का. Biomolecules में से almost 5 क्वेश्चंस पूछे गए थे इन नीट 2024 एंड आगे भी मोर और लेस इतने नंबर ऑफ क्वेश्चंस पूछे जा सकते हैं क्योंकि इसके क्वेश्चंस बनते हैं जुओलॉजी में भी और बॉटनी में भी. नाउ आउट ऑफ द एंटायर चैप्टर रिमेंबर एंजाइम्स का वेटेज 50% है. तो आज हम पढ़ने वाले हैं एक नीट बॉम्बर टॉपिक दैट इज एंजाइम्स. सो एवरीथिंग रिलेटेड टू एंजाइम्स टुडे वी आर गोइंग टू डिस्कस. जितने भी क्वेश्चंस नीट एग्जाम में बन सकते हैं वो सारे तरह के क्वेश्चंस एंड सारे तरह का कंटेंट हम आज डिस्कस करने वाले हैं. सो दैट मीन्स इट इज़ अ वेरी वेरी इंपॉर्टेंट सेशन. अगर ये सेशन आपने देख लिया और इसमें जो मैंने कॉन्सेप्ट्स बताए हैं वो आपने ध्यान से पढ़ लिए, दैट मीन्स आपके 50% क्वेश्चंस इस चैप्टर से सॉल्व हो जाएंगे. सो लेट्स बिगिन द सेशन. होप यू ऑल आर रेडी फॉर इट. सो व्हेन वी टॉक अबाउट एंजाइम्स, राइट, you have studied about catalyst in chemistry, but there is a difference between the catalyst that you study in chemistry and the enzymes that we are going to study here. तो चलो देखते हैं क्या होता है वो डिफरेंस, कितने तरह के एंजाइम्स होते हैं एंड क्या करते हैं एंजाइम्स एग्जैक्टली. सो एवरीथिंग वी आर गोइंग टू डिस्कस टुडे. Remember that enzymes are also termed as biocatalyst. तो जब हम एंजाइम्स की बात करेंगे, एंजाइम्स को बोला जाता है बायोकैटलिस्ट. So what is the meaning of biocatalyst? The catalyst that you study in chemistry that speeds up the reaction, or you rather say that it increases the velocity of the reaction. The same thing is done by our biocatalyst, right? But ये जो हमारे कैटलिस्ट हैं ये ऑर्गेनिक होते हैं एंड दे कैटलाइज द रिएक्शंस गोइंग ऑन इन द लिविंग ऑर्गेनिजम्स, राइट. सो बायो कैटलिस्ट, दैट्स व्हाई वी आर कॉलिंग देम एज बायो कैटलिस्ट, बिकॉज ये एंजाइम्स हमें मिलते हैं लिविंग ऑर्गेनिजम्स में और ये क्या करते हैं, बायोकेमिकल रिएक्शंस का कैटलिसिस करते हैं. इसका मतलब है इट इंक्रीजेज और इम्प्रूव्स द स्पीड ऑफ द रिएक्शन. Right? Now most of the biocatalyst or enzymes are protein. Remember, सारे enzymes protein नहीं है, but mostly they are proteins. Right? So सारे नहीं है तो और क्या हो सकता है, कुछ enzymes जो हैं वो RNA या ribozyme भी हो सकते हैं. So other enzymes can be ribozyme और RNA enzyme जो कि है एक न्यूक्लिक एसिड. नाउ कमिंग टू द पॉइंट कि क्या डिफरेंस है फिर कैटलिस्ट में जो हम केमिस्ट्री में पढ़ते हैं और ये वाले हमारे बायोकैटलिस्ट में या एंजाइम्स में. सो यू माइट हैव सीन दैट दोज कैटलिस्ट लाइक प्लेटिनम, पैलेडियम, दे कैन इवन वर्क एट हाई टेम्परेचर, बट since these enzymes are protein and if we are increasing the temperature they get denatured. So हमारे enzymes, these are very much sensitive to temperature, okay, because they are mostly enzymes, they will get denatured. Okay, now they are unused in the reaction. Now what is the meaning of being unused in the reaction? Let's understand. तो बिफोर स्टडिंग एंड बिफोर अंडरस्टैंडिंग कि ये अनयूज्ड कैसे होते हैं रिएक्शन में, ये समझना बहुत ज़रूरी है कि ये करते क्या है एग्जैक्टली और इसका ओवरव्यू क्या है, किस तरह से ये एक्ट करेंगे और अल्टीमेटली ये कैसे प्रोडक्ट बनाएंगे. तो रिमेंबर, व्हेन अ सबस्ट्रेट binds with the enzyme, there is formation of enzyme substrate complex, right? Now coming to the point कि ये substrate क्या है, enzyme तो हमें पता है बट ये substrate क्या होता है. So in any reaction, आप एक reaction को कैसे लिखते हो, A plus B will give us C, isn't it? और यहाँ पे enzyme act करता है. So ये जो A and B है, right, this A and B which is taking part in the reaction will be the substrate. तो आगे मैं जब भी substrate बोल रही हूँ इसका मतलब है that will be the reactant of the reaction. और ये C क्या है, this is the product. Okay. Now coming back, when substrate will bind to the enzyme, there will be formation of an enzyme substrate complex, right? Now after that, there will be formation of enzyme product complex, and ultimately you will get the enzyme back together with the product. So can you see, enzyme ने क्या किया, सबस्ट्रेट के साथ बाइंड करके प्रोडक्ट बनाया, बट एट द एंड यू आर गेटिंग द एंजाइम बैक. सो दैट्स व्हाई वी आर सेइंग दैट enzymes are unused in the reaction. Got it? So enzymes unused a reaction में at the end you are getting the enzymes back. Now this enzyme can bind to another substrate and can carry on the another reaction. Okay? So this was enzymes. Now here you can see that how the substrate are binding to the enzyme. And कहाँ पर bind करते हैं substrate enzyme के structure में? There is one site termed as active site. Active site. Okay, so active site can be a pocket or a crevice in the structure of an enzyme to which the substrate will bind. Got it? So here you can see the substrate and enzyme will be forming the enzyme substrate complex, then there will be formation of enzyme product complex, and ultimately you are getting the product together with the enzyme back. Okay, so enzyme will always remain unchanged in the reaction. Now what exactly are enzymes doing? If we understand the functioning of enzyme के enzyme actually कर क्या रहे हैं, तो ये हमने डिस्कस किया कि एंजाइम बढ़ा रहे हैं वेलोसिटी ऑफ रिएक्शन को. नाउ वेलोसिटी ऑफ रिएक्शन को कैसे बढ़ा रहे हैं, लेट्स अंडरस्टैंड. सो देयर इज अ सिंपल आंसर फॉर दिस. द आंसर इज इट reduces the activation energy. So how your enzymes are working and how your enzymes are increasing the velocity of reaction. So it is very important to write it down. Enzymes will reduce the activation energy of the reaction. Now what do we understand by activation energy? Activation energy क्या होता है? So listen to me carefully. Activation energy is the minimum energy that is required to carry on a particular reaction. तो जितने भी biological reactions हमारे body में हो रहे हैं, they will only proceed with the activation energy. Now activation energy achieve करने के लिए थोड़ा time लगता है. So if we are lowering the activation energy, what will happen? The velocity of reaction will be increased. The reactions will be faster because less time will be taken to achieve that activation energy. जब उस activation energy को achieve करने के लिए कम time लगेगा, so automatically our velocity of reaction will increase. So here is a graph. Pay attention to the graph, and if we look carefully, you will see that the x-axis is giving us an idea of progression of reaction, means how the reaction is proceeding further. The y-axis is giving us an idea of potential energy. Okay, so this is the potential energy of substrate and this is the potential energy of product. Okay, but for the substrate to convert into product, okay, there should be a transition state that should be achieved. Now ये transition state कैसे achieve होगा? By absorbing the activation energy. तो यहाँ पर आप देख सकते हैं कि activation energy without enzyme is quite high. बहुत ज़्यादा energy चाहिए होता है without enzyme to carry on a simple reaction. तो यहाँ पर आप देखो, this is the activation energy without enzyme. But with the enzyme, you can see here the activation energy is less. Yes. So what enzymes are doing? Enzymes are reducing the activation energy. Now, let's understand it by the lines of NCERT. The S, that is the substrate, has to go through a much higher energy state or a transition state. So whenever we want any substrate to convert into product, okay, so in between this process there should be a transition state that is to be achieved. Now, what is transition state? The difference in average energy content of substrate from that of this transition state is called activation energy. Now, for transition state, definitely we need activation energy. Now, remember the difference in the average energy of the substrate from that of the transition state. मतलब transition state का जो energy level है उसमें से हम अगर subtract कर देते हैं the energy, the potential energy of the substrate, what you will get will be the transition energy. Now this transition energy is more without enzyme and less with enzyme. Okay. So remember this, that enzymes brings down this energy barrier, making the transition of S to P more easy. तो क्या कर रहे हैं एंजाइम? इस ट्रांजिशन स्टेट के लिए जितना एनर्जी चाहिए होता है उसको रिड्यूस कर दे रहा है, तो इसलिए वी आर टेकिंग लेस टाइम फॉर एनी रिएक्शन. ओके, सो आई होप नाउ यू हैव understood that how an enzyme is working and how it is achieving this increased rate of reaction. Okay. Now let's talk about the structure of enzymes. So like we have discussed, enzymes are mostly proteins, but together with protein there will be some non-protein part as well in an enzyme. So there are two types of enzymes. One is simple enzymes that are made up of only proteins. Now second will be a conjugated enzyme. Now this conjugated enzymes will be made up of the apoenzyme part and the cofactor. So this apoenzyme part is the protein part and the cofactor will be the non-protein part. So this non-protein part can be of three types. Okay. So when we talk about cofactor, remember that it can be of three types. It could be a prosthetic group, which is organic, or it could be a coenzyme, which is again organic, or it could be a metal ion, that is inorganic. Okay, so that means a conjugated enzyme can be made up of apoenzyme and cofactor, and this cofactor can be a prosthetic group, coenzyme or metal ion, right? So that means in a conjugate enzyme there will be either apoenzyme and coenzyme, apoenzyme or prosthetic group, or apoenzyme and the metal ion. Okay, so it is not that all the three will be there in the enzyme, but one of it will be there. Got it? Now what is the major difference between prosthetic group and coenzymes? Let's understand. So remember always that the coenzyme will always bind loosely with the apoenzyme, or the connection with the apoenzyme of the coenzyme will be transient. So I will write it here, that coenzyme binds loosely with apoenzyme, right? Whereas the prosthetic group will bind tightly with the apoenzyme. Right? Now one more point that you need to note down for the metal ion is that it is the inorganic part that gets attached to the apoenzyme and it is forming coordinate bonds. So please write it down, very important, that metal ions will form coordinate bonds. Yes, the same coordinate bonds that you study in chemistry. So these are the major differences between prosthetic group, coenzyme and metal ion. Okay. So now let's understand the factors that affect the enzyme activity. So there will be several factors affecting enzyme activity and one such factor is temperature. So when we are increasing the temperature, remember we have discussed that enzymes are mostly proteins. So when we are increasing the temperature, they will get denatured. There will be denaturation of protein, that means our enzymes will be denatured. So mostly our enzymes get denatured when we increase the temperature more than 40 degrees Celsius, mostly. But still there will be certain enzymes which are thermostable. Okay, for example, the enzyme that we use in PCR or polymerase chain reaction. So let's write it down. Thermostable enzymes. Example of one thermostable enzyme will be Taq polymerase. We use this Taq polymerase in PCR and this enzyme is obtained from a bacteria Thermus aquaticus that live in hot water sulfur springs. So that means they can withstand high temperature. That's why we are calling it as thermostable. So let's write that also. Okay, but this is not the case when we are lowering the temperature of any reaction. So when we reduce the temperature of any reaction, apart from the optimum temperature, you will see that the enzyme will attain a temporary inactive state. Okay, so there is a difference when we are increasing the temperature and we are reducing the temperature. So our enzymes will work maximum at an optimum temperature and this optimum temperature varies for different types of enzymes. So let's write this as well. Okay. So optimum temperature will be the temperature range where the enzymes will be working to its full potential. Whenever we are increasing the temperature or decreasing the temperature, so if suppose we change the temperature by around 10 degrees Celsius on either direction, if we are increasing 10 degrees or we are reducing 10 degrees Celsius, what will happen? The rate doubles or decreases by half. Okay, for every 10 degree change in either direction. Now, when we take an example of enzyme carbonic anhydrase, so we know a reaction taking place in our RBC where carbon dioxide combines with water and there is formation of carbonic acid, right? And after that this carbonic acid breaks into H plus ion and bicarbonate ion. So this reaction is catalyzed by an enzyme carbonic anhydrase. Now this carbonic anhydrase enzyme is the fastest enzyme in our body. Now remember that a lot of this enzyme is present in the RBC, but few enzymes are also present in the plasma. Now without this enzyme, the formation of the carbonic acid will be very slow. There will be formation of around just 200 molecules in an hour. But with the help of this enzyme carbonic anhydrase, there will be formation of around 6 lakhs molecules in just 1 second. So that means this enzyme is accelerating the reaction to around 10 million times. Okay. So remember this, and now you have understood how powerful our enzymes are. Okay. Now the another one is pH. Like we have seen in temperature, that there will be one optimum temperature or the range of temperature where our enzymes will be working maximally. Similarly, there will be optimum pH as well. So our enzymes are even sensitive to pH, right? And when we increase the pH, there will be denaturation, and when we reduce the pH, there will be temporary inactive state. So here you can see that there are certain enzymes, for example pepsin, that will be working fine in acidic environment. Then comes salivary amylase, that will be fine in slightly acidic pH, and then there will be alkaline phosphatase enzyme that will work fine in alkaline pH. Okay. Now next is substrate concentration. If we plot a graph where we keep in the x-axis the substrate concentration, and on the y-axis if we keep the rate of reaction or velocity of reaction, we will get a curve like this, right? Now what this curve is trying to tell us is, when we are increasing the substrate concentration till certain extent, the rate of reaction will increase, or you can say the velocity of reaction will increase. Then Vmax will be achieved, or the maximum velocity of the reaction will be achieved. And even after that, if we keep on increasing the substrate concentration, the velocity of reaction will not increase. Okay. Now coming to the point that why this is happening. Okay. So if we are increasing the substrate concentration, our velocity of reaction should keep on increasing. No. But what will happen? There will be saturation of the enzymes. If we are increasing just the substrate concentration and not increasing the enzyme concentration, what will happen? All the enzymes that are present in the reaction will be saturated by the substrate, and there will be no more enzymes to bind with the substrate. Okay. So because of this, you will get a Vmax. Okay. Now there is something termed as Km value. So this Km value is the Michaelis-Menten constant. Now what does this means, Km value? Remember, this Km value is the substrate concentration. It is the substrate concentration at which the velocity of reaction reaches half of its maximum value. So I will repeat and try to explain you what is Km value. So it is the substrate concentration at which the velocity of reaction will be Vmax by 2. Okay. Now why this Michaelis-Menten constant is important, or Km value is important? Now because of this Km value, we will be able to understand that how efficient a particular enzyme is. So if the Km value is less, that means with just less concentration of substrate, we will achieve V max by 2 और half of the maximum velocity. तो जितना कम होगा Km वैल्यू उतना ज़्यादा एफिशिएंट एंजाइम है, बिकॉज थोड़े से ही सबस्ट्रेट में V max by 2 अचीव हो सकता है. ओके, सो रिमेंबर दिस Km वैल्यू, and the reason why just increase in the substrate concentration will not always increase the velocity of reaction. Next is enzyme concentration. The same is true for enzyme concentration as well. If you are increasing just the enzyme concentration, again after some time you will see that the velocity of reaction will not increase. So for actual increase in the velocity of reaction, you need to increase both the substrate and the enzyme concentration together. Okay. Now let's talk about inhibitors. There will be certain chemicals that can inhibit a particular enzyme and those chemicals are termed as inhibitors. Okay, तो ऐसे chemicals जो किसी भी एंजाइम के फंक्शनिंग को स्टॉप कर देते हैं उन्हें हम बोलते हैं इनहिबिटर्स. ओके, और इस प्रोसेस को हम बोलते हैं एंजाइम इनहिबिशन. सो दिस कॉम्पिटिटिव इनहिबिशन इज देयर इन योर सिलेबस. नाउ व्हाट इज अ कॉम्पिटिटिव इनहिबिशन? इमेजिन if this is an enzyme and this is the active site of an enzyme, the substrate that will fit in the active site of the enzyme, okay, will have a particular shape, okay, will have a particular shape, particular structure to fit into that active site. Imagine if there is another molecule which resembles the substrate in the shape and structure, and if it binds to the active site instead of substrate, सबस्ट्रेट के जगह पर अगर इनहिबिटर बाइंड हो जाएगा एक्टिव साइट पे, क्योंकि उसका स्ट्रक्चर एंड शेप एकदम सबस्ट्रेट के जैसा है, तो इस तरह के इनहिबिशन को हम बोलते हैं कॉम्पिटिटिव इनहिबिशन. सो यहाँ पर प्लीज राइट दिस पॉइंट दैट इन कॉम्पिटिटिव इनहिबिशन द इनहिबिटर resembles substrate. राइट, सो इनहिबिटर एकदम सबस्ट्रेट के जैसा ही होगा, साइज में, शेप में, स्ट्रक्चर में. तो एक्टिव साइट पे अगर वो बाइंड कर गया तो रिएक्शन जो है वो स्टॉप हो जाएगा क्योंकि सबस्ट्रेट बाइंड नहीं कर पाएगा. नाउ द सेकेंड इंपॉर्टेंट थिंग दैट यू नीड टू रिमेंबर इज दैट दिस इज रिवर्सिबल. ये किस तरह है का रिएक्शन है? रिवर्सिबल है. रिवर्सिबल मीन्स if you increase the concentration of substrate, this inhibition will be reversed. तो अगर इनहिबिटर का कॉन्सेंट्रेशन ज़्यादा है, एंजाइम इनहिबिशन हो जाएगा क्योंकि एक्टिव साइट पे इनहिबिटर बाइंड करेगा. बट अगर सबस्ट्रेट का कॉन्सेंट्रेशन ज़्यादा है तो सबस्ट्रेट बाइंड करेगा एक्टिव साइट पे. एक बहुत ही इंपॉर्टेंट एग्जाम्पल इसका आपको याद रखना है फॉर नीट एग्जामिनेशन, एंड दैट इज द एंजाइम सक्सिनिक डिहाइड्रोजिनेज. So in this enzyme succinic dehydrogenase, remember substrate is succinate, whereas the inhibitor is malonate. So very important example asked in NEET examination. So now I hope that competitive inhibition is clear. So if this is clear, let's understand how enzymes are classified. So enzymes are classified into 6 groups, and how you are going to remember these groups? By this small mnemonic OTHLIL. So there are 6 groups of enzymes. oxidoreductase, transferase, hydrolase, lyase, isomerase and ligase. Now based on the fact which chemical reaction is catalyzed by the enzyme, they are classified into these 6 groups. Okay. So oxidoreductase will catalyze the redox reactions. Transferase will catalyze the reactions in which there is transfer of groups. Hydrolase will catalyze the reactions where there is breaking of bond by adding water. Lyases will catalyze the reactions where there are again breaking of bond but without using water. Isomerases will catalyze the isomerism reactions, and ligase will catalyze the reaction when there is any synthesis of new molecule or there is formation of bonds. Okay. So these are the 6 group of enzymes. Now in cofactor you need to remember certain examples of coenzyme of prosthetic group and metal ions, and every year questions are repeated from here. Okay, so in one year there will be question on the example of prosthetic group, then next year there will be question on coenzymes. So like this you need to remember. Now like I told you that cofactors can be a prosthetic group, coenzyme or metal ion. So example of a prosthetic group will be heme. Now this heme may be present in enzymes like peroxidase and catalase. Okay. So prosthetic group example is heme, not the heme present in hemoglobin but the heme present in the enzymes catalase and peroxidase. Similarly, most of the coenzymes are vitamins. Okay, very important question of NEET. So coenzymes are mostly vitamins, for example niacin. So in NAD, NADP you have niacin vitamin which is vitamin B3. An example of metal ion is zinc in the enzyme carboxypeptidase. So carboxypeptidase is a proteolytic enzyme and in that enzyme the metal ion that will be present will be zinc. Okay. So with these examples of cofactor we have completed all the important points of the enzymes. Okay. So remember, I am reminding you again that enzymes will hold around 50% weightage in the chapter biomolecules. Okay. And remember the concepts that we have discussed now, that will be helpful for your preparation and definitely you will improve your marks. Thank you very much everyone. See you soon.